thank you very much. I'm very grateful to be able to share my my work with you here in Hong Kong. It's really great to be here and also to open the conference. Yes. So my work title is From Objectual to Explanatory Understanding with Alphafold2. This is not working. Yes, right. So some information on our project first. Maybe I will just move this to a corner where it doesn't. Yeah, right. So to start, I work in a project comprised by junior professor Florian Boge, me and our PhD student Annika Schuster, who will also present later. Yeah, right. And we are working on scientific understanding in the context of deep neural networks. And so very quickly, just an overview. So we have identified 3 foreseeable paths to scientific understanding in these fields. The first one is to just explain the deep neural network itself and to uncover its discovery method. The second one would be to match deep neural networks predictions with an explanatory model afterwards. And the third one would be to use the deep neural network to understand facts and dependencies in the world directly without understanding the deep neural network itself. And so this third path is the one that I am looking at because it's being done in science a lot, and in particular with the case of Alphafold2. So a very why this is not working? Yeah, right. So to give you a very short introduction, because I don't know your backgrounds, what the protein folding problem that Alphafold2 is supposed to tackle. So you might remember from biology classes that proteins are synthesized in the ribosomes from messenger RNA and comprised of amino acids that form polypeptide chains. And the interesting thing, or one interesting thing about proteins is that they come in very different shapes. As here's just a selection of proteins to illustrate this. Each protein has its own unique shape, and connected to their shape is their function in the body. So this is very important information for scientists. And right, so proteins play a vital role in the body. They are concerned with catalyzing chemical reactions, with transport, and signal transmission, for example. And there are hundreds of millions of known amino acid sequences that correspond to proteins, but only before AlphaFold, there were only 200,000 known structures that were experimentally determined, which is a very difficult procedure that takes a lot of effort and sometimes even 10 years for one single protein. So there was there's this huge gap between known sequences and structures. So what is the protein folding problem, or why is there a problem? Why can't this be predicted? So there are 3 questions related to this. The first one is the thermodynamic question that asks, what is the specific balance of interatomic forces that dictates a protein's native state, which is the folded state? The third one is kinetic. What routes or pathways do proteins use to fold in so quickly as they do? Because they fold in a very short amount of time, while the space of potential conformations that they could take is very big. And the third one is computational. So how can this 3D structure be predicted from the amino acid sequence? And then there was the entry of Alphafold2. So DeepMind released Alphafold2 in 2020, and there was a lot of excitement about this, and only recently also the Nobel Prize was awarded to one of its developers. So and there were things like headlines asking, has DeepMind's AlphaFold solved the protein folding problem? And there was this general spirit like, okay, now we did it. Now we solved the protein folding problem that's been around for more than 50 years and really puzzling. And you can also see huge rise in research since, especially since July 2022, when DeepMind released the source code of Alphafold. So, right, so very many publications on the topic of Alphafold. So, right. So what we are asking ourselves in the project is, right, so Alphafold is a deep neural network. It's a transformer network. So it's not really clear how exactly it's arriving at the single predictions that it's making. Still, scientists seem to think that they understood something about protein biology. So what is this and how can we make sense out of this? So to tackle this question, I created like 3 criteria for scientific understanding with a method that I will talk you through in the beginning. Then I will briefly discuss the problem of understanding with black boxes, and in the last step, then combine the two and talk about understanding with Alphafold2. So in the debate on scientific understanding and philosophy of science, there has long been a received view according to which understanding is just a psychological byproduct of an explanation and as such not directly relevant for science. But in the last, I think, 20 years, people began to notice that understanding equips scientists with important skills, like skills you need to really create models or to make the right idealizations. It's not enough to just know an explanation. You really have to understand what you are doing. And in this context, the concept of objectual understanding was also devised, which refers to the understanding of a phenomenon or of a discipline, like somebody understands evolution or understands protein biology, which you can gain via explanations directly, but also from understanding or from grasping dependency relations, from obtaining certain skills, or from unification. Some people like Lipton and Geisbers argue that objectual understanding can even be had without an explanation, or some others say that this kind of understanding is reducible to explanations in the end. In my work, I assume that all methods that contribute to either objectual or explanatory understanding are important for scientists, because so if a DNN could provide us with any of that, it would be a great starting point. While I leave out, I will not take a stance on this debate. So my criteria can be visualized in a very idealized scientific process like this. So I just assume, and I know that it's a lot more complicated in real life, but I assume that the scientific process has at least a phase of exploration, a phase of hypothesis generation and testing, and a phase of theory construction and refinement. And so in the phase of exploration, where I put my integration and coherence criterion, it is important to observe nature, of course, to collect data and to analyze data. And in this context, deep neural networks are used as a method to collect data and to analyze data. The second criterion is the potential explanation criterion, which refers to this second phase in the idealized process, where we generate potential explanations and hypotheses, derive predictions and test predictions. And the third phase, or the third criterion, is the explanation criterion, which comprises the construction of explanations for a phenomenon. Right. And so objectual understanding and explanatory understanding can be located in this scientific process as well. Right. So my first criterion, the explanation criterion, reads like this: M contributes to the scientific understanding of P if scientists use it to develop a scientific explanation of P. So this is pretty difficult to achieve because usually a method's output requires additional scientific analysis, but Lawler argues that there are model explanations where the non-propositional model itself is the explanation. You can think about the plate tectonics model, for example, from which you can directly, without even like making it propositional, see why earthquakes might arrive, just from understanding the plate tectonics model. And the second example would be the Lotka-Volterra model. My second criterion is the potential explanation criterion. A method M contributes to scientific understanding if scientists use it to develop potential explanations for P, for the phenomenon. So this would refer to all methods that allow scientists to develop how possibly explanations, hypotheses, and answers to what-if questions. And this all is valuable objectual understanding, which can lead to explanatory understanding, as I will argue later. And the third criterion is the integration and coherence criterion, where the method contributes to the scientific understanding of a phenomenon if it is used to gain new information on the phenomenon that raises the coherence of the body of knowledge of that phenomenon. So this is all that understanding that comes from methods that provide data or data analyses, and understanding that comes from understanding the phenomenon or the domain surrounding it better itself. So just from gathering more data, we can arguably understand a phenomenon better, even if we don't interpret them at all. There's a very nice talk by Stephen Grimm on this topic, where he makes this the first level of understanding, to just understand the phenomenon. Right. So the second part of the introduction is understanding with black boxes. I borrowed a graph here from my Florian. Right. So when we are talking about understanding with black box models, we might be talking about two different things, actually. So if we imagine we have a scientist, we have a phenomenon in the world, and we have a black box machine learning model, then we could ask how is the machine learning model working, but we could also look at the predictions that this machine learning model is working and ask about the phenomenon. So directly talking about the phenomenon without understanding the black box directly. And my question is, do researchers understand proteins or protein folding better with these Alphafold2 predictions? So, and the argument would be that to understand phenomena, a deep understanding of the vehicle might not be necessary. This goes back to Duran in 2018, where he says that, or where he makes a case for that understanding the workings of a computer is not necessary to understand something with a computer, generally, although there are exceptions sometimes. And so the philosophical debate has two important contributions that I want to mention. The first one is by Emily Sullivan in 2019, where she develops the concept of link uncertainty. And she says that models only explain to the extent that there is a link that connects the model to the phenomenon. And this link has to be established with additional scientific evidence that supports this connection between causes and dependencies of the model and causes and dependencies in the target phenomenon. She says the main challenge is whether the various implementation black boxes that prevent us from explaining and understanding certain aspects about how the model works also prohibit explaining and understanding the phenomenon the model bears on. So according to Sullivan, some understanding with deep neural networks seems to be possible if we embed them right in the scientific process. Rätz and Weisbart are less optimistic. They say that the predictive success of deep neural networks is a brute contingent fact and not something that we understand theoretically. This in turn threatens our understanding of phenomena when we apply deep neural networks in science. And they say that ML models can, however, lead to some objectual understanding, for example, by establishing correlations or by simply adding to knowledge of a domain of things. So what I want to ask is, couldn't this weak objectual understanding, so they make it out to be a bit weak, lead to explanatory understanding in the course of the scientific process? But of course, so they would agree that machine learning models can fulfill my integration and coherence criterion, right? So on to understanding with Alphafold2. So Alphafold2, this is the structure. I will not go into details because it's not important for my talk. What is important is, so we have Alphafold2 takes as an input the amino acid sequence or any amino acid sequence, and its output is a complete 3D structure prediction of this amino acid sequence, and it is using multiple sequence alignment, MSA representations, which is evolutionary information that has been used in protein biology since a long time, because it's known that, for example, if fish and human have a similar protein, then they might have, then these proteins will have a similar shape, at least in the parts where they are similar. So the multiple sequence alignment is comparing the structures for different organisms. Then, right. It also uses templates if they are available, and it uses pair representations, but I will not go too much into detail. Just what I want to show with the slide is that it's not using any laws of protein folding or any laws surrounding protein folding, like the hydrophobic effect or anything. I will go into more detail. So, for example, the assumptions made in Alphafold2, the first one is that the amino acid sequence is cut into triangles based on the backbone structure of amino acids, which is why they refer to it as a gas of rigid 3D bodies, which is, of course, biologically implausible because, yeah, they are polypeptide chains, which are, right, which have a bond between them, right? But yeah, Alphafold2 imagines them as triangles, or a gas of triangles. But it has been thought that this is actually very useful for Alphafold2 to detect long-range dependencies, which, for example, if the blue and the yellow amino acid in this schematic representation do have some interaction, maybe because of some attraction between the molecules, it would be much easier to find if you imagine them as free triangles, right? The second thing is that there's the absence of typical cell conditions like water, pH value, or the temperature in Alphafold2, which is known to play a very important role in protein folding. For example, there's this rule that hydrophobic side chains usually fold to the center of the protein, and the hydrophilic side chains stay on the outside because they are soft in water, right? And so this is missing from Alphafold2, all this information, and there's also the absence of functionally important molecules, because sometimes proteins bind, for example, iron, which has an important plays an important role in the function of the protein, but is not part of the amino acid sequence. And also Alphafold2 does not show any dynamics. For example, there are proteins that regularly occur in two different conformations, but Alphafold2 only outputs one of those. So this for the problems, although to be fair, so this seems biologically, if I say it like this, it seems biologically not very plausible. However, Alphafold2 was trained on all the available proteins from the protein database, so trained on almost 200,000 structures. And of course, all these structures reflect the presence of water. They reflect that proteins are not triangles, and they reflect also these important molecules. So it's all inside the training data, and some argue that Alphafold2 has in fact found some biological mechanisms. So what are some possible targets of understanding with Alphafold2? So the first one would be the protein folding problem directly. How can we understand from Alphafold2 how proteins fold in general? And here, I would say this is not possible because first, it makes biologically implausible assumptions, and second, there's no dynamics in the predictions. We get a static structure as the output in the end, but this does not tackle two of the important questions, if you remember, like how do they fold so quickly, and how which balance of interatomic forces is determining the folding. The second question is a bit more modest. It's, can we learn something about how the protein universe is structured or clustered? Like we have a useful amount of predictions, which is, all right, I didn't mention this, but we went from 200,000 known structures to 200 million known structures with Alphafold2 in the Alphafold database. So it's a very impressive amount of predictions. But a certain uncertainty and the dynamics behind proteins play a role here. But to give one example of a very interesting research paper where they tried to incorporate the information from Alphafold2 is by Bordin et al. In 2023. They used the Cas database, which is a database that identifies domains and protein structures and classifies them into evolutionary superfamilies, and thereby provides structural and functional annotations. So they added the Alphafold database data, so 200 million proteins, although they had to exclude some. So this expanded it considerably and provides valuable insights on structure-function relationships, as they say. So as you can see, for example, with the inclusion of the Alphafold database proteins, we have an expansion in the class 1, evolutionary class 1, which looked not as important before, but seems to dominate in the second picture. And a reduction in the class 3, for example. So some analyses like this just become possible because there are a lot more protein structures available now. Right. So a quote from their paper is: the expansion and structural diversity in Cas superfamilies brought by Alphafold2 relatives is exciting, as it could help rationalize functional divergence in these superfamilies. Right. And so I would argue that the integration and coherence criterion is fulfilled here. The Alphafold2 prediction contributes information on the distribution of structures in the protein universe. Right. A second interesting example comes from the research on nuclear pore complexes. Those are very complicated proteins found in the cell, at the border of the cell nucleus, right? And human nuclear pore complexes are beyond the largest known protein complexes, with over 1,000 proteins taken from multiple copies from a set of about 30 different nucleoporines, NUPs. So we have two kinds of NUPs, the scaffold NUPs that contain folded domains and form a cylindrical scaffold around the channel. And we have the intrinsically disordered NUPs, which line the scaffold and interact with cargo complexes. Right. So Mosala-Ganti et al. In 2022, they made an analysis of the structure of this nuclear pore complex. And this is a quote: to gain a better molecular understanding of NPC function. So they directly mention better understanding. And they used Alphafold2 in this. Right. So what they did was they checked for all the different nucleoporines in the Alphafold database, and they were all available and had a high confidence score. In a second step, they compared them to X-ray images that have been that were unpublished at this point in time, and found an astonishing similarity between the two, which led them to conclude that they can use these predictions and fit them into an electron microscopic map. And so in this case, they really used Alphafold2 predictions to obtain these hypotheses about the fitting of different nucleoporines into these images. Because it's very difficult to get good images of the inside of this pore. So in this case, I would say that they fulfill the potential explanation criterion, because Alphafold2 predictions really helped in constructing possible explanations for how these proteins fit inside the pore. And the resulting models cover various domains and interfaces that so far remain structurally uncharacterized. So this was really very important work. And they say that the completeness of our model has enabled microsecond-long coarse-grained molecular dynamic simulations of this scaffold within an explicit membrane environment and solvent. So while the contribution from Alphafold2 prediction was only objectual understanding in the form of a hypothesis, this contribution allowed the researchers to deeper understand the mechanism, and thus to devise an explanation and fulfill the explanation criterion. So what I sometimes title a new pathway. So my conclusion is that we only get objectual understanding from Alphafold2 predictions, but predictions plus the background knowledge of scientists plus some exploration can lead to explanatory understanding in the end, if scientists are embedded in the process. Right. So to conclude, or for a quick outlook, I say that Alphafold2 predictions provide this new objectual understanding of the protein universe, and they serve as very good templates that facilitate and accelerate experimental verification considerably. The enhanced objectual understanding then, combined with standard scientific practice, such as computer simulations, plausibly contributes to the solution of protein folding for single proteins. But the protein folding problem itself has not been solved, but rather avoided, or has become less important because we can get very good predictions without understanding how proteins fold in general. Right. So thank you for your attention.
Okay, any questions? Mario. Hi, very interesting talk. Could I ask what would qualify for you as a solution of the protein folding problem? Would you need a mathematical formula, or what is it?
Right. So it's actually, if we look back at the formulation of the protein folding problem, it's an answer to 3 questions, right? So the thermodynamic one would probably look somewhat like some formulas that describe how interatomic forces dictate this native state. The kinetic one, usually possible explanations or theories that are looked at the moment look something like this, that you have some energetic landscape. But how this landscape is exactly determined is unclear. So you would need some solution to that. And yeah, the computational one would kind of follow from the other 2. Good.
Could I follow up on the second? I'm not sure that the second question, that I fully understand the second part, because this seems like an abstract picture which has nothing to do with reality. In reality, there is not such a landscape. It's a mathematical model. So why should you talk about the dynamics in an abstract landscape that you might use to get some intuition, but there's nothing to do with what is actually going on in nature?
Well, I'm not sure I agree. Well, it's you could certainly argue that this is part of reality. I mean, these are some calculations that show you how the status of Gibbs free energy of different possible conformations, right? It's pretty difficult to get good data on how proteins behave because they fold very quickly, they are small, and it's very difficult to capture images of them. So, but sometimes researchers get these images too. So, yeah, I would say this is maybe more real than you say. It corresponds to something that's there in reality, at least, yeah, possible conformations.
Okay, so this is about the dynamics. Then I think I misunderstood you, and then I agree with you. Okay.
Okay.
Thanks.
Thank you.
Thanks. Just to follow up on that, I mean, you just mentioned Gibbs free energy. Actually, of course, answering there's more than one question in one. I mean, because the interatomic forces is different from working out the correct minimum energy state, right? So I just would want to say that there's more than 3 questions. I think there are, in fact, a multitude of questions that you could ask, and you could answer some without answering others.
Oh, right.
Would you agree?
I'm sure that's true. Yes, yeah. Right. So these are probably summaries of more fine-grained questions. Right. Yeah. Yes.
Absolutely. No, the reason I think it's interesting is because, say, there could be something like a representation hidden in Alphafold2 that could do one of those things, like the free energy thing, but it can't do the forces. In fact, I suspect it couldn't do the forces for sure.
Exactly. Yes.
There's nothing in the data that would give you that, I think.
Yes, there's actually a very interesting paper on this by some people who tried to find this thing, and they basically say that Alphafold2 found a good way to find a starting point to navigate this landscape, which is something that researchers really need, because if they just they don't know how to start, and that's why they don't find the right.
Yeah, Andre mentioned this. We should talk about this afterwards.
Yes.
Thank you. Hi, thanks for a good talk. I was going to say good morning to you in the elevator earlier, but it's been a bit too much of a guess, I guess. The question I have concerns the impact of sort of attitudes of scientists towards whether the problem has been solved or not. So you said, scientists can now move on to other things. And I wondered if you'd looked at something like, now there's more papers to do with AlphaFold. Are there less papers to do with, say, ab initio calculations to try and solve, to really solve the protein folding problem, if you like? Because 20 years ago when I was at university, people did lots of ab initio calculations to try and model the forces for very, very simple molecules even. Right. And I wonder if there's now, less interest in that, maybe less funding as well, even over time.
Right. I didn't exactly look into this, but I think these concern different kinds of researchers still, right? So researchers who are really interested in solving the protein folding problem, they probably don't use Alphafold2 much. But many people depend on the results, right? So there's so many, like in medicine, for example, they really need to know how certain proteins fold. And also for protein design, it's very useful to have something that predicts you actually almost any structure, right? So I would think that I think it's possible that there's less funding. It's even plausible that there's less funding for research because it's lost some of its appeal. But it's still, researchers always make clear that the Alphafold2 predictions are often a good starting point for research, but they can never be taken at face value completely. So just putting your amino acid sequence into Alphafold2 does not give you enough reliability to really work with that. So in addition, they always do an experimental verification. The point is more that it's becoming a lot easier to get, because it's a lot easier to verify a structure when you have a good template. And Alphafold2 is giving mostly good templates, although there are also some problems. I didn't go into detail. That's very interesting, but yeah.
Thank you very much.
Yes.
Well, thank you very much for the great talk. So I have one clarificatory question. So can you remind me about the difference between the objectual explanation and the deeper explanation?
Right. So explanatory understanding is basically understanding that derives from grasping an explanation, right? So when you grasp an explanation, you get explanatory understanding. Objectual understanding is much broader and also refers not only to a single phenomenon, but more to a whole discipline. Like you can understand why rainbows form, like why a rainbow is on the sky at the sky right now, right? And the answer would be an explanation that you grasp. But you could also understand rainbows, and this would not only comprise this question, but also a lot of different questions, like maybe some relationships between angles of sunlight, some things that might not be necessarily explanatory relevant, but that are still in the bound, like within the phenomenon, like something that helps you understand rainbows without being directly relevant to the explanation.
I see.
Right.
So then I have a kind of two questions. One is that is the difference the kind of difference in kind that they are completely different, or in degree, that is if you kind of obtain more and more objectual understanding, we get at some point you gain the kind of explanatory understanding. That's one point. And the next question is that, so your conclusion is that, well, AI can give you a kind of objectual understanding, but you have to add some kind of background information and so on in order to get the explanatory understanding. Do you think that it's possible in the near future, or maybe future, that machines can give us an explanatory understanding by itself?
Interesting, thank you. The first question, so this is debated actually, and it's a debate that I don't want to go into because it's very, it's a debate mostly in epistemology, right? And I'm more on the philosophy of science side of things, but right. So some people say objectual understanding is broader than explanatory understanding because it comprises explanations, but also a lot more information. While other people say, like Khalifa, for example, he says that, okay, all the objectual understanding is basically explanatory understanding, and we only need explanatory understanding. Objectual understanding is nothing. So we have two camps there, basically. And I think they both have good arguments. I don't have time to go into this debate, so I take it that both are relevant. And they are, yeah, for this, there are good arguments, I think, right? And yeah, I think that would be great if AI could do this in the future. Right now, it doesn't seem to be really, there doesn't seem to be so much motivation to really, I mean, there is some motivation to make AI explainable, of course, but this kind of precision that we would need to really like understand, for example, Alphafold2 predictions, well, it would basically have to tell us a whole story about why, why does this protein look like this, right? So to really get an answer, yeah, I mean, the question was, do you think it's possible that AI does this?
For example, ChatGPT seems to kind of answer the why question. For example, when I ask something why in physics or something, they answer, they try to answer it, right? So
yes,
we have the same kind of similar thing.
Yeah, I think that's very exciting. I would hope so, but I don't. Yeah.
Okay. Well, thank you.
Thanks. Yeah, I think, isn't philosophy of science the true epistemology?
Yeah, well, when I said that, I regretted it directly. Please don't take me up on that, okay?
Thanks for the talk. Are you able to go back to the slide where you talk about understanding the distribution of structures?
Yes.
I just had a question about that.
Oh, yeah, very good. Okay, yeah.
Yeah, so I guess I was kind of wondering how much understanding we really get here, because like, as you noted, the vast majority of these aren't going to be sort of independently validated, like we can't sort of validate them experimentally. AlphaFold is kind of known to perform poorly for certain kinds of proteins. And there's kind of this general worry that, you know, when you have ultra fast AI systems like this, you can generate a lot of slop.
Right.
And so I wonder if you need to like add some additional conditions on sort of like the goodness of the predictions that you're getting or something like that. Because otherwise it might look like you're kind of like actually increasing the amount of like misinformation about the, you know, distribution of structures as opposed to the information, if the predictions aren't especially good. So yeah, could you say something about that maybe?
I mean, we talked about this a few weeks ago, and since then I'm thinking about this. Yeah, actually, right. I see this with different eyes now. Like, it's true that I'm also skeptical now, although I think, so these are evolutionary superclasses, right? So this is already a level of abstraction that's pretty high. It's basically, I'm not a protein biologist, but it depends on the how many alpha helices you have, how many beta strands you have. So it's on a pretty general level, and this is what Alphafold2 seems to get mostly right. So I think there's at least some value in this, but it's definitely true that as they are not externally validated, all the 200 million structures that are in the Alphafold database, it is, yeah, a healthy amount of skepticism around these results is definitely, yeah, valid. And also the related point that you made, that this might actually pose a problem for protein biologists who are really interested in this, because it makes it costs them a lot of time to just go through everything to really see what is the quality now. We cannot, yeah, right. Like with machine learning models in general, they produce vast amounts of data, right? So it costs a lot of time to just validate them and go through them. So this is really a problem that, yeah, I don't have it in the talk, but I'm working on that, and I will have it in the paper. Yes.
Thanks. I'll ask a question or two. I should stay on that topic for a minute. I mean, the laws it finds and the relationships it finds can be really ugly and approximate and only even maybe work for some particular proper subset of all the molecules it can consider, but they can still be laws, I think, right? I mean, there may be the kind of laws that we haven't in the past used much in science because we've been focused on, you know, having very simple things that are easy to remember. But it seems to me that when you think about empirical laws, there can be loads of them and they can be very complex. So yeah, I wouldn't be. And what do you think? That wouldn't put nothing about what you said would put me off the idea that it could find, Andre, that it could find, you could find some kind of laws in this way of that kind, right?
Yeah, so what Roney et al. Did was definitely to something like this, and I also think there's probably something hidden. Problem is just that it's hidden. So So
I wanted also to ask about two other questions. One was about what you said about whether any laws are built into Alphafold2, and you mentioned the triangle. Were you talking about the invariant point module there? The invariant point. What's the IPA? It's called. My IPA. When I hear IPA, I always think of pale ale, and I can't think of the invariant point association. So that's to do with working out the structure. That seems to me to be a physical law to do with how things can occupy space, right? So it's a mathematical relationship, but that is a physical law that's built in, I think. Would you agree?
Yeah, I mean, chemical, physical, yes.
Yeah, so yes. I think so. I just think there are some things built into Alphafold2 that help it a little bit.
Yes.
That maybe in some other cases that aren't actually. I've been thinking about GraphCast more recently, which is really interesting because there's no physics built in at all, and it still does a really good job.
Yeah.
The other question I wanted to ask was about the pathways at the beginning. I wanted to just make sure I understood the difference between pathway 2 and pathway 3.
Ah, right. Yeah.
Because I thought that sometimes doing one could do the other, but I think they probably do come apart.
Probably, yeah. Well, pathway 2 is like we match deep neural networks predictions with an explanatory model, right? So this would mean we use the deep learning predictions that we have, and then we kind of post hoc make sense out of it, and we devise a model afterwards. Whereas in path 3, we just completely ignore the opacity, basically, and we say we use it to directly understand facts and dependencies of the world, right? So
okay. Yeah. I mean, I guess maybe I was thinking the two could come together quite closely, because if you're not matching all the predictions, you're not copying the whole algorithm, you're just copying the predictions in one context, then maybe the distinction can sometimes collapse.
That's a good point. Yes.
Okay. Yeah. That's what I was thinking.
Right.
Great. Any other questions at all? Sure.
What you just said about pathway 2. So before AlphaFold, there was of course AlphaGo that did all sorts of. And I have, like by chance, I happened to play the game, and I watched a few. I watched maybe 100 or so analyses by top pros of AlphaGo playing against itself, and I was sort of struck by the idea, by the sort of, here's a professional player trying to presented with lots of data and trying to make sense and trying to give an explanation for what he was seeing, and it was quite complex sort of hypothesis building, sort of looking down paths that weren't taken in the game to try and say, this seems like an obvious move, but the computer did this, and here's a sort of 20 move long-winded potential explanation for it. And I wonder if that fits into your path 2 model and how they feel about it. And they didn't seem that certain about whether they'd figured out the real reason, if you like, for the moves, which might be an interesting analogy to explore.
Oh yes, I think that's interesting. So there's also a paper on where chess players try to comment on new routes. There's a really interesting paper. Yes. No, but right. So I would think that this would fit as an explanatory model, right? Yes, if they try to make sense of it. Yeah. So they match the predictions with the model. Yeah.
Good. Yeah. Thanks a lot. The AlphaGo one's really interesting because sometimes knowing about something about the way it works and what the modules do is important, right? So it seems the reason that Lee Sedol actually beat it was because it judged in that particular game that there was a very low probability of a human playing the move that Lee Sedol played, a God move it's been referred to, and that's partly why it made the move, just because it thought, oh, I mean, he can win, but he won't see that, so to speak. Great. Let me thank you very much again for your talk.
Thank you very much for the discussion.